So total capillary blood flow equals cardiac output. So it's determined by the pressure and the resistance in the cardiovascular system. If there isn't sufficient pressure and the resistance is too high, then you're not going to get capillary blood flow. This occurs to people who are in intensive care, and they can get what is called disseminated intravascular clotting, where they are so ill that we cannot control their blood pressure and we cannot control the resistance with all the chemicals that we're trying to give them, and in the end stages of their life, what happens is that the capillaries start to clot off. So here you can see just an overview of what controls the circulation. So if we start up at the top, we've got cardiac output, and we're going to move to the right following the arrows around. So we have arterial blood pressure. Within the artery we have peripheral resistance, so that is determined by how wide the actual artery is, and if there's anything impeding inside the artery, like atheromas with coronary artery disease. Then we move down to the capillary pressure. Remember that there are different types of capillaries. We need a certain amount of pressure in order for the fluids and the nutrients and the gases to be able to move out of that capillary bed into the interstitial fluids. After it's moved out of the interstitial fluids, it will move back through the venules to the venous pressure, and this is all controlled by the middle bit here, which is under neural and hormonal control. So neural means nerve control, and hormones, remember, they're the endocrine organs. So what you can see there is the little kidney bean shaped, of course, is a kidney, and on top of it, the little triangle one, what looks like a little hat, is the adrenal gland. So the adrenal gland, it tells you that it actually secretes adrenaline, which is a sympathetic hormone which has a powerful effect on the cardiovascular, causing effect on the blood vessels. Then we move back with the venous return back to the heart. So pressure is shown here as P, and the heart generates pressure to overcome resistance. And the resistance is caused by all of the blood vessels. So that's called circulatory pressure. And it's the difference between the pressure at the heart and the pressure at the peripheral capillary beds. So flow is proportional to the difference in the pressure gradient divided by the resistance. So measuring blood pressure, we measure blood pressure via a non-invasive method where we wrap a cuff around the person's upper arm usually, and what we do is we pump up the air in the bladder around the patient's arm and compress the brachial artery against the humerus bone. Then we slowly and we pump that up until we can until we completely obliterate the artery and close it off momentarily. Then we undo the little cuff tap very slowly, carefully. That's the tricky bit. And then listen very carefully with our stethoscope. And when we hear the very first beat of the heart coming through, the first squirt of blood that's coming through, that is equivalent to the arterial pressure, measured in millimeters of mercury. Another type of pressure is the capillary hydrostatic pressure. So this is the pressure within the capillary beds. The capillary beds need to have a sufficient amount of pressure to be able to move the solutes and the gases out of the blood vessels into the interstitial space. And then you have venous pressure, and this pressure is the pressure required for the rest of the venous system with blood that is travelling back to the heart against gravity. So the gradient pressure across the systemic circuit is about 100 millimeters of mercury. So the circulation pressure must overcome total peripheral resistance, the resistance of the entire cardiovascular system. So vascular resistance can be created due to friction between blood and the blood vessels, and it depends upon the vessel length and the vessel diameter. So adult vessel length is constant, the vessel diameter varies between vasodilation and vasoconstriction, and the resistance increases exponentially as the vessel diameter decreases. So the smaller the vessel, the more resistance it is. So viscosity is resistance caused by molecules suspended in materials in resistance is caused by molecules and suspended materials in a liquid. So whole blood viscosity is about 4 times that of water. So if we took a test on a piece of glass and we had the piece of glass on an angle and we were able to put a drop of blood and a drop of water, and then we lifted that piece of glass until the water started to run, the water would beat the blood down to the bottom of the glass at a ratio of about 4 times. So it would take the blood 4 times as long to run to the bottom because of all of the formed elements within the blood. Here we've got a diagram, well, a series of diagrams that are explaining what the slides did previously. So if we talk about friction and vessel length, so here we've got 2 blood vessels. One is half as long as the other, and you can see that the resistance in the top one is 1 and the flow is 1. The resistance in the second one, because it is twice as long, is 2, and the flow is half as much. If we looking at an illustration of friction and vessel diameter, you can see that the least resistance is right in the middle of the vessel, where you've got blood just running against blood and blood not being caught up against the sides of the vessels. The final diagram that we're looking at in the box is vessel length versus vessel diameter. So diameter is how from one side of the inside to the other is 2 centimeters, and then the one underneath it you can see is half the size, that's 1 centimeter. So the vessel length versus vessel diameter, so here we've got the same length, but the diameter of one is half of the other. So in the diameter, which is 2 centimeters, the resistance of the flow is 1. If we then halve that diameter and take it down to 1 centimeter, remember it said that the resistance is exponential. So here we have a resistance that is equal to 16. So it'll take 16 times as much force to get the same amount of fluid through. And then in the blood vessels that are diseased, you've got plaque deposits. Remember that is atheroma, so deposits of fat such as cholesterol inside the tunica media, pushing into the diameter on all sides of the blood vessel, and of course that will interrupt how and increase the resistance of blood flow. So it will take more blood to push through diseased vessels, which is why patients who've got coronary artery disease often have hypertension or high blood pressure as well. So these next series of diagrams are taken from your book, and we'll just talk through what we're looking at. So here you can see on the x-axis where we have the vessel diameter in centimeters, and on the y-axis we've got the different types of blood vessels in the human body. And we're discussing vessel diameter. So you can see here that the biggest diameter is in the aorta, and then you also come across to the vena cava, which is bigger again. But in between that, we've got the muscular arteries, the arterioles, the capillaries, the venules, and the veins. You can see that the diameter decreases until you get down to the capillaries, and then on the venule side it begins to increase again. Here we're looking at the cross-sectional area in centimeters squared. So you can see that the capillaries have by far the greatest cross-sectional area, which is very important because it's there where all of the nutrients and the oxygen, the carbon dioxide are exchanged with all of the living tissues. So we need as much as possible the sectional area, cross-sectional area, to be the greatest in the capillaries. In this diagram, we're looking at average blood pressure. So you can see that Again, we've got the same order of blood vessels, where we've got the aorta, the muscular arteries, the arterioles, the capillaries, the venules, the veins, and the vena cava. So blood pressure is highest when it leaves the left ventricle and goes into the aorta, and blood pressure is lowest when it returns via the superior and the inferior vena cava to the right atrium of the heart. And you can see that there is sufficient blood pressure at the capillaries for what was that hydrostatic pressure to make sure that the nutrients move out of the capillary beds into the interstitial space. This particular diagram, it looks at the velocity of blood flow. So as we would expect, the aorta has got the highest flow of blood, and we have the lowest flow of blood at the capillaries. So we've got blood moving through all the time, but it is a lot slower at the capillaries. Again, that is so that there is time for the nutrients, oxygens, and also the waste products to exchange. And you can see the vena cava. Also, we've got blood moving rapidly, but under less pressure. So arterial blood pressure, the systolic pressure is the peak arterial pressure during ventricular systole. Diastolic is the minimum arterial pressure during diastole. All right, so now we have the understanding of what blood pressure is. Remember, we have the two readings, the systolic over the top of the diastolic. So have normal and abnormal blood pressure. So the normal range for blood pressure for an adult, we're looking at about 120 over 80. Hypertension, so hyper increased blood pressure tension is known as hypertension, and according to the National Heart Foundation, that we generally agree that anybody who has got a sustained blood pressure, resting blood pressure of 140 over 90 is said to have hypertension. And it indicates blood vessel disease. May also indicate too much fluid, like fluid overload. The next one is hypotension, so low blood pressure, so abnormally low blood pressure. And this can be caused by not taking sufficient fluid in, but also elite athletes have a lower blood pressure because their level of fitness, so that when they are at the highest peak of their sport during their endurance, then their blood pressure isn't dangerously high. Elastic rebound, the arterial walls can stretch during systole, and then they can rebound to their original place, original shape during diastole, and it keeps blood moving during diastole. Venous pressure and venous return. Low venous resistance is assisted by muscle compression of peripheral veins. So the compression of skeletal muscles pushes blood one way with the one-way valves, which is what we spoke about in the previous lecture, the valves of the veins. But there's also what is called the respiratory pump. So the thoracic cavity has an action. During inhaling and exhaling, there are pressure drops within the thoracic cavity. We'll talk about those next week and in week 13. But as the person inhales, there is a drop of thoracic pressure, and exhaling, there is a rise in thoracic pressure, and this helps bring blood back to the heart. Capillary pressure and capillary exchange is vital for homeostasis. So it moves vessels, it moves materials across the capillary walls by three processes: diffusion, filtration, and reabsorption. So we'll look at these in a bit more detail. Remember that diffusion is the movement of ions or molecules from a high concentration to a low concentration along the concentration gradient. So diffusion relates to ions or molecules moving down the concentration gradient. Filtration is driven by hydrostatic pressure. So water and small solutes are forced out through the capillary wall because of pressure, and it leaves the larger solutes in the bloodstream. So larger Larger solutes include larger proteins and of course the formed elements of blood. We don't want the red blood cells and the white blood cells unnecessarily, well, we don't want red blood cells moving out of the capillary walls. We want to keep all that going around in the circulation. Then you've got reabsorption, and this is the result of osmotic pressure. Remember that osmosis is the movement of water, and this is in relation to blood colloidal osmotic pressure. I'm just introducing this concept to you. You don't need to know more than this about it. You'll be talking about it more in next semester and in second and third year. So equals pressure required to prevent osmosis. So the blood colloidal osmotic pressure has to be high enough to prevent water moving out of the extracellular fluid into the vascular compartment. And this is caused by suspended blood proteins that are too large to move across the capillary walls. So what we're looking at here is the diagrams of what we've just spoken about. So in the top part of the diagram you can see the pink vessel wall with endothelial cells with a little nucleus, and you can see that the vessel wall has got some small holes through it, and we've got amino acids, blood proteins and glucose, and you've got the little small green dots which are ions, and so that's in the vessel wall. And then underneath it you've got the interstitial fluid. Remember, that's the fluid that the cells are bathed in, so it's not inside the cells. And what we're looking at, if we look at that little square box down below, we can see here that we've got small solutes that are able to move through these small compartments. So that's how some nutrients move through the capillary beds and into the interstitial space and then into the cells. So there's an interplay between filtration and reabsorption that ensures that plasma and interstitial fluid are in constant communication and mutual exchange. And it does accelerate the distribution of nutrients, hormones and dissolved gases through the tissues when things are all working well. So the capillary exchange at the arterial end of the capillary fluid, so we're talking about the arterial end, so this is where the fluid, the blood is high in oxygen and high in nutrients. Here we want the capillary exchange, we want fluids to move out of the capillaries into the interstitial space, and then at the venous end where we want fluids to move back into the capillary, because we want things to move out of the interstitial space, interstitial fluids, and we want to move back into the capillary. So these would be the waste products and carbon dioxide. So here you can see a diagram of what we've been talking about. So you've got on the left of the diagram, you've got the arteriole, and you can see the blood flow from the red arrow to the blue arrow, and this is capillary plexus, where we've got a small capillary which goes from being artery area of high in oxygen and high in nutrients to an area of low in oxygen, high in carbon dioxide. So you don't need to remember this diagram, but you do need to understand the concept that we have fluids moving in and out, and with it we've got nutrients moving in and out. It all works to maintain homeostasis and make sure that every tiny cell in our body receives the nutrients, and waste products are removed every second of our lives.